session this evening. Kristen, you're on.
Wonderful. Good evening, everyone. I'm Kristen Kokeman, President and CEO of the ALS Association Greater New York Chapter. Thank you so much for joining us for our Education and Resource Summit and this evening's session covering advancements in ALS research, presented by Dr. Kuldip Dave and Dr. Neil Shneider. Dr. Dave is Vice President of Research for the ALS Association and oversees the development and implementation of the global research vision, including funding strategy, research programs, and strategic initiatives. Dr. Dave received his bachelor's degree in biology from Rutgers University and his PhD degree in pharmacology and physiology from Drexel University College of Medicine in Philadelphia. Prior to joining the ALS Association, Dr. Dave was with the Michael J. Fox Foundation for Parkinson's Research. Dr. Shneider is the director of the ALS Association Certified Treatment Center of Excellence at the Eleanor and Lou Gehrig ALS Center at Columbia University, where he oversees an integrated ALS clinical care and translational research program. He is a graduate of Harvard College and of the program of the MD PhD program of the Columbia University College of Physicians and Surgeons, and completed his residency training in the Harvard Longwood Neurology Training Program. More information on both speakers can be found in the Speaker Center on the Summit platform. Tonight's research presentation will run for approximately 90 minutes and will include a question and answer session. Please use the chat function to send in your questions, and they will be presented to Drs. Dave and Shneider. If we are unable to get to your question, we will respond offline, and there will also be another opportunity on Friday during our closing session for follow-up questions. I'll now turn the discussion over to Drs. Dave and Shneider to get us started.
Kristen, just give me a thumbs up if you see my screen come up.
You are all set. I can see your screen. It looks good.
All right, great. Thank you. Whoops, sorry about that. Thank you to Kristen and the New York chapter for this invitation, and I look forward to this session with Dr. Shneider. What I thought I would do is go through our overall strategic pipeline for what the ALS research ecosystem looks like, and then walk you through some of our projects that we're funding and how they fit into this pipeline. So with that, I want to first remind you that, while I'm going to talk about research today, it's important to understand that we have a three-pronged mission. We have a mission to serve and care for and advocate and discover treatments and a cure for ALS. And that happens through these three prongs of our mission. Care services, from which we have a network of chapters and clinics all across this country that provide care and support programs and resources to both patients and family members, and thereby enhancing quality of life and support for families that are affected by this devastating disease. We also have an advocacy mission, and by that we work through focusing on educating and mobilizing members of Congress, and do so by increasing government funding for ALS research and accelerating drug approvals. And we help change laws and policies that benefit the ALS community. Today, I'll talk about what's in the middle here, which is the research and the department that I lead at the ALS Association. And as you know, we are the largest worldwide grant program that funds research projects, research infrastructure projects, and research training projects. We provide funding to academic labs, to pharma and biotech industry, and really our goal is to accelerate the most promising and significant advances in ALS. So with that, I'll talk about this ecosystem, and I know this looks very complex, and there's a lot of things here. So don't worry. I'm going to walk you through this very slowly from the left all the way to the right. So, and if there's anything that you take away, I want you to take away this picture, this ecosystem, because if you understand this, you'll understand what's happening in the research ecosystem today. So starting all the way on your left, I don't know if you can see my cursor here, but on the left here, that says in red, biology and genetics of ALS. This is really the very first step in this ecosystem. It's the most important step because this is where we try to understand what is different in ALS, what is dysfunctional in ALS, what goes wrong in ALS, biologically, in our cells, in our muscles, in our neurons, in our bodies. And this is really important because this is where we understand what targets are different, and those targets are what we can now make therapies against. And so understanding what kind of genetic alterations may lead to increased risk of ALS. Underneath that is risks and causes of ALS. And what I mean here is beyond the genetic risks, what are the environmental risks? What are the occupational risks that may change your risk of getting ALS? For example, are there particular environmental factors or chemical pollutants that increase your risk of ALS? Or is there an occupational risk? For example, we know that people who have served in the military have a twofold higher risk of getting ALS. What are the things? It's important to understand what is it about the military service, or what is it about living in a particular environment or being around particular industry that may increase your risk of ALS. And so that research is really important. After you go from this initial stage, you get into the next stage, the green stage. And so let's assume we found a target, either through biology or genetics or an environmental factor that's affecting something in our cells. That's the target. Now you start making drugs against that target. And that's what happens in this green stage, the preclinical drug discovery stage. This is where you now start doing chemistry, or if you're making a genetic therapeutic, you start developing a biological therapeutic. This is where this mostly happens, while the first part here on the left mostly happens in academic labs. This part here mostly happens in industry, in small biotech, in pharma. And they are doing chemistry and lead optimization and screening. And these are all drug discovery terms that you don't need to know about. The point is that we're making drugs and we're testing it in animals, whether they work in animals, whether they get to the brain and spinal cord in animals, whether they're safe in animals. That's what's happening in this preclinical drug discovery stage. And underneath that is biomarkers development. Biomarkers are objective measures of that allows you to see whether they may help you diagnose the disease. They may help you see if somebody will progress faster or slower. They may even help us select populations for particular clinical trials. And so biomarkers are very important, and they also fall in this middle phase. And once you've shown that a drug is safe in animals, now you take it into humans. And that's this phase 1, phase 2, phase 3. So phase 1 is where you test it in healthy individuals or in people with ALS, whether the drug is safe. Phase 2, now you start to ask questions about, does the drug work? Does it get into the brain or spinal cord? Does it what doses may be more effective, which populations may be more effective. That's what's happening in phase 2. And then in phase 3, those are much larger trials. You look at how the drug behaves in a larger population. And now, now that pipeline happens and a drug goes through phase 3, you get into the real world where a drug is approved. But even on that side, there's a lot of research that can be done. You could do research in phase 4 studies, which means what's the drug doing in the real world? There is research about assistive technology. There's research on telehealth, telemedicine. There's research we could do on families and caregiver burden. We could do research on access and quality of care, research on natural history of the disease, and of course, patient surveys. And these things are really important parts of research because this research is happening in the real world with patients who are providing that those data to us. And then underneath it all is this piece called infrastructure, which allows all of this to happen. And research infrastructure is very important because that infrastructure is really a fancy word for capacity building. We need to build capacities in labs, whether they're doing preclinical work, whether they're doing clinical work, whether that's specimens, data collection, tools, training of fellows, or we want new people in the ALS space. And so how do we train them to be in the ALS space? And so all of that is really important. And just in case you're wondering what the ALS Association funds, we fund the entire part of that ecosystem. There are aspects, and I'll go through this in a later slide, different projects that touch different elements within this ecosystem. Just to give you a sense of our current commitment, we are funding 162 active projects at this time, across 13 different countries. And if you add up the amount of dollars we have committed to those 162 projects, it comes out to $55 million. That's our current research commitment in the ALS space. By the way, this number changes all the time. Just a month ago, this was up at 168. 2 months ago, it was at 172. It'll go back up once our new funding programs are launched and contracted. Point is, this number is changing, but our overall research commitment at this stage is $55 million. So what are the challenges here? And the challenges are many for neuroscience drug discovery. Again, I'm showing you this pipeline here. And as you walk through this pipeline, as somebody goes through this pipeline, discovering a target that's different or a mechanism that's different, developing drugs against it, testing it in animals, and then testing it in humans, all the way up to drug approval. This process is very time consuming. It can take an upwards of 10 years to get something through this process. We know this because think about SOD1 gene that was discovered in the late 90s and early 2000s. And just now we're seeing trials with SOD1 like tofersen that are in phase 3. And so the point is, it takes a long time to get through this stage. This is just a complex pipeline, and this is just how science works. Not just time, it takes a lot of money to go through this. There are reports that it could take greater than $1 billion to get a drug to go all the way from preclinical target ID and target validation into preclinical drug discovery and into clinical testing to get a drug to the market. And unfortunately, even with the time we spend and the money we spend, the failure rate is very high. Right now, it's greater than 95% of drugs fail going through this. They may fail at the preclinical stage. Maybe they're not safe in rats. They may fail in phase 1 where they're not safe early on, or maybe they fail in phase 3 where they're not efficacious or they don't have significant effects over placebo. And so the drug discovery pipeline is complex, it's slow, it's costly, it's risky. And so our strategy our strategy needs to be so that we are maximizing this pipeline. We are developing this pipeline as diversified as possible, so that when drugs fail, there is something standing behind it that's going through this pipeline and ready to go through this pipeline. And so it's important that our research feeds this pipeline constantly, so that when things fail, we have something in the back that's coming along and hopefully will be positive. And so and by the way, this is not, this issue is not just common to ALS. It's actually all of the other neuroscience diseases have the same issues. I come from the Parkinson's field, and it's exactly the same. So it's not a matter of how much money you're spending. Michael J. Fox Foundation spends $100 million each year, and there is no cure for Parkinson's. Alzheimer's Association spends more than $100 million for Alzheimer's each year, and there's no cure for Alzheimer's. So this is not an issue of the amount of money; it's the issue of being smart, and with knowing that this pipeline is complex and neuroscience drug discovery is not easy. So what are our research operating principles? And this is really our strategy. We fund and invest. Since the Ice Bucket Challenge, the ALS Association has committed over $113 million to research. And we will continue to fund and invest in new ideas and across that pipeline. And I'm going to show you examples. I'm going to walk you through that pipeline and show you an example within that ecosystem of what we have funded. We collaborate and partner. And this is really important. This is a space where we cannot afford to have the walls up or be in silos. We need to collaborate. We need to partner, and we do that. Organizations in the ALS space do that all the time. For example, we have partnered with Project ALS, another nonprofit organization, and to fund earlier stage JC Fusen story that Dr. Shneider will mention later. We have collaborated with Target ALS, which is another nonprofit organization, to develop biomarkers for TDP-43 measurements in fluids. You may have seen a recent press release where we collaborated with MDA and ALS Finding a Cure to fund the stem cell, the T cell therapy that Dr. Pell down in Houston is developing. So it's really important to collaborate and partner in such instances. We convene and connect. It's important as the largest nonprofit organization in this disease that we bring people together. For example, last year in October, we had a workshop where we brought together worldwide experts to talk about environmental risk factors and occupational risk factors. And what will it take for us to take an environmental risk factor and provide guidelines to the community? And so it's important that connection and convening of experts is really important, because that's where ideas can be shared, where new funding ideas can come up. We lead and advocate, and this is again really important as the leader and someone who stewards and sets the agenda in the ALS space. We need to lead, and here an example of this is genetic testing guidelines, and Dr. Shneider can talk about this, but there are no guidelines right now as to who should get tested genetically. If you have a family history, if you don't have a family history, there's really no guidelines for ALS specialists. And so what that means is there's inequity in how people get access to genetic tests. So we have taken the leadership in that and are working with a couple of investigators to develop guidelines for genetic testing and counseling. And that's what we mean by leadership. And we leverage and de-risk. And really, our dollars can go so far. So our goal is to fund at a critical juncture, critical stage, where then we can allow our funding and the results of our funding to then investigators to go on and get larger funding, whether it's from the government or whether they partner up with venture capitalists, or whether they get funding from pharma or biotech. And again, Dr. Shneider will touch on this. An example of this is how our early funding for the JC Fusion has now led to the announcement that just came out today from Ionis that is going to follow up on that trial and going to invest in a worldwide trial for developing therapeutics against the FUS gene. And so this is really our research strategy. Whoops. So you may wonder, how do projects get selected? How do we, you saw, I told you earlier, we have 162, 163 active projects, but we have a lot of projects that have, closed over the years. How do projects get selected? In a very rigorous, transparent, and deliberative process. So I'm going to walk you through this process so that you get an understanding of how this happens. Each and every one of our projects goes through this process. There is no exceptions. The very first thing that happens is a letter of intent. We ask investigators to send us a short proposal, a short pitch. In fact, Dr. Shneider, who is you're going to hear from next, when we funded Dr. Shneider for the JC Fusion study, we asked Dr. Shneider to send us an elevator pitch, a 1 to 2 page idea. Tell us what your idea is. What do you want to do? Once that letter of intent comes in, we look at it, we see that it's a priority, it's the idea, whether it's novel or something that Dr. Shneider is retesting, whether it fits the scope of our funding, whether it fits under our strategic planning. And if it does, we ask the investigator to submit a full proposal. And we ask Dr. Shneider to submit now a longer proposal, 8 to 12 pages. And this proposal is now peer-reviewed by external subject matter experts. And again, this is really important because we want external subject matter experts, people who may have done those types of studies, to look at the full proposal submitted by Dr. Shneider and review it, score it, provide critiques. And these experts are looking at it. They're looking at the scientific plan, they're looking at team, they're looking at budget, they're looking at the feasibility. Is this doable in the time and the money that Dr. Shneider is proposing the work? And they score these proposals. Now, this review committee will only score the full proposal. They are not providing the final approval. They're only recommending what should be funded, whether it should be funded or not. The final approval is given by the next stage here, which is the research committee. And the research committee is the research committee of our board. And these are volunteers that are comprised of patients, caregivers, former caregivers, scientists, clinicians, business professionals. And it's the research committee that provides the final approval for funding and budget. So I want you to understand how rigorous and the 3-stage process there is before a grant becomes, before a proposal becomes a grant. And then what happens at the end? So in green, you see when something becomes a grant project, we don't provide the full funding right up front. So if Dr. Shneider got a $200,000 grant, he would not get $200,000 up front. He would get $50,000 up front. And then at 6 months, Dr. Shneider would submit a progress report, which would be reviewed. And if Dr. Shneider is meeting the milestones of the project, we would release the next payment of $50K. Same thing happens at 12 months. Same thing happens at 18 months. And this is how we are able to make sure that we steward the funding and the donations that we raise from you, and make sure that the science that is funded and the work is that it happens in the right way. In addition to these milestone-based payments, we provide we ask that our investigators collaborate with each other. They share data and specimens with each other, and they publish that data so that so that others can learn from that, and we can build on that idea. Okay, so now that I went through this process, and I hope you understand and appreciate the rigor that goes into selecting, let me take you back to this pipeline, and let me go through each and every part of this pipeline, give you some ideas of the type of projects we're funding. Now, again, I'm giving you one example of each of these elements. Okay, I'm not talking about all 162 projects. I don't want you to think that this is all we're funding. We're funding 162, 163 projects, $55 million commitment. But what I'm going to do is walk you through some examples here of what we have funded. So in the biology side, we're funding Dr. Matt Harms at Columbia in New York. And here's an example of the initiative called GTAC, or Genomic Translation for ALS Care. And Dr. Harms is really doing gene sequencing to understand what kind of genetic alterations are there and how can they be related or linked to the disease. And you may know, well, by the way, this is not the only gene sequencing initiative that we've funded. These are, by the way, very expensive initiatives. This initiative itself was over $3.5 million. But the investment in these initiatives, we funded this, we funded Project Mind, we funded Answer ALS, we funded Create Consortium. The idea behind these large gene sequencing initiatives has bore fruit because now we have over 25 genes that have been shown to be linked to the disease. And so that is an important thing, because those targets, those gene targets that we find can become therapeutic targets of tomorrow. For example, SOD1 gene and C9 gene are now in phase 2 and phase 3 testing. And so the more genetic alterations we can find, the more targets we'll have for therapeutic intervention. We're also funding toxin exposure. For example, we're funding a couple of studies right now where people are trying to look at exposure to chemical pollutants, such as organic solvents or industry waste or super sites, and people that live around these exposures, to try to see exposures in the air or the water, exposures in the body, and link it to ALS diagnosis. The CDC registry also funds a lot of work around this type of work. But again, this is important to understand how toxins may be driving ALS incidents in certain regions of the world. Preclinical drug discovery. Here, I'll give you an example. Talk about funding something really, really novel. We last year funded a preclinical drug discovery project that was looking at a live biotherapeutic product. So this is not a chemical. We are funding a lot of other companies that are doing chemicals. This company in particular in California is actually giving microbiome or bacterial type administrations to see if they can change the gut microbiome. And by changing the gut microbiome, you change inflammation in your gut, and thus inflammation in your blood, and thus inflammation in your brain and spinal cord, and potentially have a therapeutic effect. That is extremely novel and a project like that. We are funding, this year, sorry. I forget that we're already in 2021. We funded a study in Israel that's looking at microRNAs. These are small RNAs that are found circulating in our body, in our CSF and biofluids. And the idea that perhaps microRNAs can predict whether a patient is going to advance faster with the disease or advance slower with the disease. And that would be really important for someone like Dr. Shneider, who is treating these patients. And so if we can find a progression biomarker like that, it would really change the way these and treatments are given out. We are funding a phase 1 study, for example. RNS-60 is a study that's being done in Europe with this antioxidant drug that's looking at whether RNS60 is safe in ALS patients. We're funding a phase 2 study. I mean, we're funding the Healy platform trial. We, over $3 million for 3 years. And as you all know, the Healy platform trial is a way to test therapies quickly and all at the same time using the same infrastructure. So again, the idea here is to make sure that we can keep that pipeline flowing through and accelerating through quickly. Like every other nonprofit, we're not able to fund a full phase 3 study. There's just no nonprofit that does that. Even someone the size of Michael J. Fox, where I worked before, didn't fund phase 3 studies. But what nonprofits do is that we fund sort of ancillary or satellite studies in phase 3, because those are normally smaller, something we're able to fund, and that helps the community. And now, in this case, our funding to the Neuron study last year to measure biomarkers in their phase 3 Neuron study that Brainstorm did is an example of that. We are currently funding on this side of the equation, on the managing ALS side, we're funding a research study that's looking at sleep in young caregivers. So let's say a mom or a dad has ALS and their kids, young caregivers, teenagers, or early 20s, are taking care, or caregivers. And what happens in that case to those caregivers? What kind of stress and burden do they take on due to a family member having ALS? We have our ALS Focus survey platform, which if you don't know about, you should definitely look into it. This is your voice. We're collecting your voice, not voice as in voice, but you are telling us what the disease is doing to you, or and we're collecting that information so you can be part of that clinical trial, even if you're not part of any other clinical trial. It's a study where we collect your information. I told you earlier about the genetic testing guidelines. We fund fellowships. We give preclinical fellowships or medical fellowships. Again, we want new brain and new people coming into the ALS field. And so if we can fund fellowships, we can do that. And we provide funding to the NEALS, which is the largest consortium for ALS and runs almost all clinical trials in ALS in the US. And we provide funding for their infrastructure and trial site training. So again, this is just an example that I gave you of the different stages of the ecosystem and how diverse our funding is and the types of projects that we're funding. This is not everything. This is just a taste of how we try to maximize this ecosystem. I'm not going to talk about this a lot, the clinical trial landscape, because my colleague, Dr. Jill Yorsak, and Dr. Jinsey Andrews from Columbia is going to talk about tomorrow. So that's something to look forward to, the clinical trials. But just wanted to show you, and she's going to show you probably the same slide. There is a lot of activity right now in the clinical space. There's over 100 clinical trials, both interventional and observational, all across the world. And this is not a full exhaustive list. This is just giving you some ideas. And the point I want you to take out of this slide is there are small molecule compounds here, and there are genetic therapies here. There are large pharma, and there are small companies. There are stem cell treatments, and there are antibody treatment. The point is that it's an extremely diversified portfolio currently, and it's a really exciting place to be in, because we want, the more these phases are populated, the more chances of success we're going to have. The more shots on goal we have, the more chances of success we have. I think my time is running out, so maybe I'll try to wrap it up in 5 minutes. But the point here is, this is just telling you how our advocacy is so important. Our advocacy is important because when we can advocate for more money, the more money federal funding is going to come into the ALS space. And so our advocacy department has been making sure that we get more and more funding for the Department of Defense program, for the NIH. And you can see this starting 26 into 2021 this year. And of course, 2021 is sort of projected. This is not a complete year yet. So our funding and the funding from the other agencies is still to be determined. But the point I want to make is look at 2016. If you take our funding and the funding from the CDC and the NIH and the Department of Defense, we were around $80 million. Today, this year, we think we're going to be double of that at $160, $180 million. And so we can't just think about the ALS Association funding. We have to think about funding from the federal agencies. We need this type of funding to increase. And so, over the next 3 years, our advocacy team has made it their mission to increase this 180 million to close to 250 million, $250 million. And if we can get that funding into ALS space, that would be amazing. The other strategy you have to remember is, yes, there's so much I can do as the head of research at ALS Association and directing dollars, but there's a lot I can do by being involved with other funders. So, for example, we partner. I spoke about this. Collaborate with other nonprofits. I attend meetings that MDA has, or ALS Finding a Cure has, or Project ALS has, so that we work with them and match our dollars so that they go further. I sit on the ALS Investment Fund Advisory Committee. I sit on pharma companies steering committees, so I can steer their dollars and their strategies towards ALS. I sit on the Department of Defense's programmatic panel, so that's a $40 million program, a lot bigger than ours. And I can, by sitting on that programmatic panel, I can have a voice and integrate the patient voice into what that panel does strategically and from their investment dollars. And I sit on the foundations committee for the Healy trial, which is a $100 million study. And by sitting on that committee, again, I can have a voice and bring my perspective and the perspective of the ALS field to that trial. So this is my last slide, and I'll end in a minute. But I just wanted to let you know that the state of the field is exciting and diverse and robust right now. We have a diverse set of targets, biological targets. But no, let me start here in blue first. We are greater and greater increased understanding of ALS, whether that's through epidemiological studies, that's through gene sequencing, that's through omics. I talked about Dr. Harms's GTEC initiative. We are finding more and more targets. We're starting to understand ALS, this very complex disease. We have many different targets. We have many different pathways, inflammation, protein handling that we are going after. Many different drug approaches. I just talked about that, small molecules, antibodies, antivirals, Antisense, stem cell approaches. We have engagement from different institutions. There's over 150 drug companies that are currently have a preclinical or a clinical program in ALS. And that's really exciting. We have clinical trial efficiency, such as what you see with the Healy platform. The FDA put out a guidance last year for trialists so that they know what kind of trials they should design in ALS. And we have a community that's engaged. You are joining us today. You're engaged. You're listening to us. We have other programs like ALS Focus, where you are providing your voice into the survey program. And so again, I'll stop here, but I'll say this is a really exciting stage that we're sitting at, and at a cusp of, I hope, a discovery for cure and treatment for ALS. With that, thank you for the opportunity. And I'm going to now stop sharing here and ask Dr. Neil Shneider to take this presentation forward. Thank you.
Thank you, Dr. Dave. I'm going to share my screen here. Can you see that? questions. I'm really pleased to join you tonight. Thank you, Kristen. Thanks to the New York chapter of the ALS Association for putting together this evening. Kristen asked me to talk in this first night of a series of talks about advancements in ALS research. This is a huge topic, of course, nothing that I think I could deal with in 20 or 30 minutes, and I want to leave plenty of time for questions tonight. But I thought I would just sort of give a general overview of the field and some of the exciting advances in the field, including some clinical trials that have been mentioned by Dr. Dave and Kristen, that I'll spend some time talking about to give you a sort of a sense of the process, expanding a little bit on what Dr. Dave has told you. Beginning, of course, with the disease itself, amyotrophic lateral sclerosis was first described in 1869, over 150 years ago, by Jean-Martin Charcot in Paris, and it came to the attention of Americans pretty late in 1940, when Lou Gehrig, the famous Yankee, was diagnosed with this disease. And I think before that, people really hadn't heard of ALS, didn't know what it was. That still may be the case largely, but I think it's a fair question to ask, really, what has changed in the 80 years since Lou Gehrig died of ALS? How has our ability to treat this disease, to alter its natural history, changed? Some might say critically that it hasn't changed all that much, that our ability to manage the symptoms of the disease and to help our patients through the course of the disease has improved significantly, and it has. But yet there are only 2 FDA-approved drugs at this moment, and both have, I would say, modest but significant effects, but are neither of which, and both together, are not enough, and we really need to do better. But I share Dr. Dave's enthusiasm and excitement about this moment. We've learned a great deal in the 80 years since Lou Gehrig came to public attention. And I just wanted to give a very, I think, limited, maybe superficial sense of what we've learned in that time. You know, part of the problem for ALS and for all neurodegenerative diseases is that these are complex disorders. The nervous system itself is incredibly complex. ALS is a disease that affects multiple cell populations throughout the nervous system, from literally top to bottom. This, I don't know if you hope you can see my cursor, but ALS involves neurons in the motor cortex that project down into the brainstem, into the spinal cord, to activate a second neuron, a lower, so-called lower motor neuron that innervates the muscle. And the degeneration of both of these neuronal subtypes leads to the muscle weakness that leads to the signs and symptoms of ALS. And if this wasn't complicated enough, in addition to the motor problems that are associated with ALS is related very closely, clinically, and as I'll show you, genetically and pathologically, to another neurodegenerative disease called frontotemporal dementia, or FTD. And this is something that our field has realized more and more in recent years, of the relationship between ALS and FTD. And many of our patients, you know, fall on a spectrum where they have some degree of frontotemporal dysfunction, and in some cases, upwards of 15% of our patients have frank frontotemporal dementia. So all of our patients are on the spectrum that I've depicted here, where some have pure motor neuron disease, have ALS, others have pure frontotemporal dementia, but many of the patients that we see in clinic have some degree of both motor neuron disease and frontotemporal dementia. ALS is the most common adult onset motor neuron disease. It affects about 5,000 to 6,000 people every year. That's the annual incidence in our country. The lifetime risk is pretty high, surprisingly high, when people hear this, that as our life expectancy increases, 1 in 350 men and 1 400 women will develop ALS. The disease course is still on average 3 to 5 years, and it affects people in the 5th and 6th decade of life. The large majority of our patients, over 85 to 90% of our patients, have what we call sporadic disease. That is, that they come into our clinic and they have no obvious family history. But about 10% of cases, the disease is familial. It's due to a genetic mutation that's passed down from one generation to the next. And as you'll see tonight, a great deal, maybe a disproportionate degree of attention is spent on the familial cases. And there's some criticism from the community. Why is it? The familial cases only represent 10% of all cases. Why is so much attention paid to this minority of ALS patients? And I'll make the point again and again tonight that what we learn from the familial cases is relevant to the large majority of the patients we see in clinic, whom we cannot distinguish clinically. That is, when a patient comes in to see me, if they don't tell me about their family history, I cannot, based on their examination and their history, I cannot distinguish a familial case from a sporadic case. There are important links between the familial and the sporadic cases that I think justify our focus, the focus of the field, particularly in recent years, on these familial cases. So it really, I think, begins with the question of what causes ALS. And Dr. Dave mentioned a few possibilities. There have been many that have been suggested. Viral or infectious causes have been suggested, inflammatory, autoimmune problems have been suggested as causes of ALS, toxic environmental causes. None of these have been proven. The only known cause of ALS, the only thing that we know is responsible for the development of ALS are genes, genetic mutations that cause familial ALS. But as you'll see, these mutations in these same genes that are responsible for the familial disease are also found in patients who have no family history for a number of reasons. So our focus on genetics really makes possible a cycle of discovery that begins with our patients. And as Dr. Dave described, allows through the study of our patients to understand more about the genetic architecture, the genomics of ALS. The discovery of gene mutations allows for the creation of models, animal and cellular models of disease that we can then take into the laboratory and study to discover mechanisms that underlie neurodegeneration in ALS and FTD. And in these pathways, we find therapeutic targets, which we can then go after for drug and therapeutic development and bring these therapies back into the clinic for the benefit of our patients, completing the cycle. ALS is a genetically complex disease. The history of discovery in ALS began in the early 90s with the discovery by Dr. Robert Brown, and Tipu Siddique and their colleagues of mutations in the gene SOD1, superoxide dismutase. And over the last 30 years, there's been this absolute remarkable period of discovery of new ALS genes in which mutations cause ALS, and in many cases also frontotemporal dementia. And you can see that I like this figure because each gene is represented as a circle. The size of the circle represents the relative incidence. How common is that mutation in familial cases? And you can see that, and the calendar years is on the y-axis on the left here. And you can see that the slope of this line is decreasing. That means the pace of new gene discovery has accelerated dramatically over the last 10 to 20 years, owing to the publication of the human genome and to the discovery and advancement of next generation sequencing technologies and computational methods to analyze that data. This has led to really an extraordinary period of gene discovery in ALS that has fueled our development of models, our study of disease mechanisms, and the therapeutic discoveries that are now emerging. So I won't spend much time on this, just to say that in familial cases, in about 2/3 of people who have familial disease, we now can identify the specific genetic cause of their disease. And importantly, these mutations in these very same genes are also responsible for about 8 to 12%, let's say, of sporadic cases of ALS. Genetic mutations causing disease in people who don't know that they have a family history for any number of reasons, which I think makes the point that Dr. Dave alluded to earlier of that it's important, we believe, to offer genetic counseling and testing to people even without a family history, given the possibility of discovering one of these causal genes in our patients. So the hope of ALS, genetics was we would study our familial cases, that we would get a small number discover a small number of genes, and they would identify a single pathway or a small number of pathways that would point us in the direction of cause and suggest a therapeutic strategy that we could use to help all patients. Well, that's not how it worked out. Out of the dozens of genes now that have been causally associated with ALS, we find that they can be categorized, they can be put into multiple different boxes, multiple different functional categories. And this has created a lot of confusion, if you will, but difficulty in our field. How is it that these disparate genes doing multiple different things in cells and in our nervous system, how is it that these mutations all cause this very stereotyped disorder of motor neuron degeneration in the brain and spinal cord? How do these pathways, how do these genes relate to one another in a way that makes sense? And how do we then approach these disorders? The group of genes that I think, to me anyway, are most interesting and where there's been a great deal of focus in the last 10 plus years are on a group of genes that encode for RNA binding proteins involved in RNA metabolism in the synthesis of RNAs and their processing in the cells. Two of those genes are important, and I'll mention them repeatedly: TDP43 and FUS, fused sarcoma. So the important point about TDP43 was many, but the important part is that TDP43 is a major component of these large inclusions, these large abnormal structures within the body of the motor neuron. Here's the nucleus of the cell, and here's this large inclusion that stains positive for TDP43. There are also mutations in the TDP43 gene that cause ALS. But what's remarkable is that TDP43 pathology, absent mutation, is found in 97%, in the vast majority of cases of ALS, show this kind of TDP43 pathology, and that's true of sporadic cases, and it's true of many familial cases, and it makes the point that again that familial forms of ALS and sporadic forms of ALS are related. This is a pathological similarity between the two that suggests that what is going wrong in the neurons of sporadic ALS patients is also going wrong in our familial cases in most instances, and that the underlying mechanisms responsible for the neurodegeneration are the same. Therefore, a therapeutic that works in one case, familial case, may well work in another, and vice versa. So I'm going to spend just a minute, I think, on this, going back to this slide here. The largest circle in this slide is that which represents mutations in the gene called C9orf72. This was a genetic mutation that was hidden in a way and discovered only in 2011 by Rosa Rademakers and Brian Traynor and their colleagues. And what they found was that in this gene, there was a sequence, 6 nucleotides, GG, GG, CC, that is normally repeated just a handful of times in normal individuals. But in ALS and also in frontotemporal dementia, this expansion is massive. It's repeated hundreds, if not thousands of times. And subsequent studies have shown that this is the most common cause of ALS and frontotemporal dementia that we know. And a great deal of effort, remarkable effort in just 10 years, has identified a number of possible mechanisms by which this expansion might cause neurodegeneration. One of them is through a loss of function of the C9orf72 gene. That is, it has a critical function, and when you interrupt that function through this expansion, you get a loss of function that leads to neurodegeneration. It's one possibility that's being pursued. But 2 others involve a gain of function. That is, that abnormalities, that this expansion causes the mutant RNA or its product, but the mutant RNA to cause problems in the neuron and to lead to neurodegeneration. And that could be through the presence of the expansion itself, which might bind critical elements, critical factors that are important for neuronal survival. But it's also been found that this sequence, this GGCC, actually makes proteins, abnormal proteins that are toxic to the cell. So there's a lot of effort to better understand the mechanisms of the disease. But one approach that's been taken to deal with this most common cause of ALS and frontotemporal dementia is rather simple. The RNA is causing a problem, so eliminate the RNA. And what has developed in recent years, thanks to efforts of companies like Ionis, pharmaceuticals, which I'll tell you more about. They and other companies, many now in this space, have developed technologies to use small pieces of DNA, in some cases now RNA, as sort of smart bombs that they can go in with a specific sequence and target an individual gene product for destruction. And that is the approach that was taken for the case of C9orf72, but it was really based in many ways on previous successful efforts in a related form of motor neuron disease called spinal muscular atrophy. Ionis, in partnership with Biogen, developed, really, I think, one of the most exciting therapeutics in neurology in recent years, a small antisense oligonucleotide, a small piece of DNA, which alters the expression of the gene SMN2, which is responsible for SMA. This is a antisense therapeutic that is now in the market, FDA approved and used in the treatment of children with spinal muscular atrophy. A similar approach has now been taken for C9orf72 related forms of ALS, and the oligonucleotide, the ASO, originally developed by Ionis, now in partnership with Biogen, has moved this forward into a phase 1/2 multiple ascending dose trial, and this is a trial that is active right now in our center and many centers around the world, trying to use this technology to eliminate the C9orf72 RNA, the expanded RNA, in a way that would slow and hopefully halt progression of disease. A similar approach, similar technology with an ASO now specific to the SOD1 gene is in now phase 3 clinical trial. Dr. Dave mentioned this earlier. It's a similar approach. We understand, based on our study of animal and cellular models of disease and study of patients with SOD1 mutations, that the mutations in the SOD1 cause that protein to become toxic to neurons. So one way to eliminate that toxicity is to eliminate the offending RNA and its product, the SOD1 protein. This is the drug called tofersen, and the trial is shown presented some exciting phase 2 data and is now in phase 3, and we're waiting anxiously to know whether this drug has efficacy in slowing, halting, and maybe even reversing some of the functional decline in patients, ALS patients with the SOD1 mutation. Another trial has recently been initiated, again, an Ionis Antisense molecule developed in partnership with Biogen. This Ataxin-2 ASO, that is an antisense molecule that is targeting the Ataxin-2 gene, is very exciting because this is one that could potentially benefit patients without a causal ALS mutation. Ataxin-2, based on some very brilliant preclinical work, has been identified as a modifier of the toxicity in ALS, and that the idea is that by altering the levels of the Ataxin-2 RNA, you can modify or improve the disease course for patients. And this again has, I think, great promise for a majority of ALS patients with sporadic disease, that is without a known causal mutation. And this is, I think, one example, and there'll be many more in years to come, of this kind of an approach using this technology that's now focused on the familial forms of the disease, that will be applied to the sporadic patients and people without causal mutations in an effort to alter the natural history of their disease. And this brings me to the fusion study, which Dr. Dave mentioned earlier. This is an antisense approach to patients with mutations in the gene FUS, fused in sarcoma. This is a study, as I'll tell you about, that developed out of my laboratory. It began in part when we met a young woman named J.C. Hermstead. She had a mutation in the FUS gene that caused ALS. She had an identical twin sister named Alex, who had, of course, the same mutation and died of complications of ALS at age 17, when she first became symptomatic at 11 or 12 years of age. So we were interested in helping JC. We turned to Ionis to ask them if they had a potential ASO therapeutic that might be used to help JC. They were remarkably responsive. They offered us access to the FUS ASOs that they had in development. And we were able, with the help of Project ALS and with the ALS Association, to organize a first-in-human study of this FUS ASO, which we dubbed JC-Fusin in JC's honor. JC was the first of our patients, but then again, with Project ALS and ALS Association support, we were able to continue our work, and now we've treated 12 patients, with this antisense oligonucleotide under an expanded access, or otherwise known as compassionate use protocol. And we were able to advance this therapeutic in the clinic with Ionis's attention and support. And this led ultimately to the phase 3 trial that was announced today called Fusion, that will offer this oligonucleotide, this antisense therapeutic, to FUS ALS patients around the globe in an effort to demonstrate its safety and efficacy, and hopefully bring this to patients, albeit a small minority of patients with FUS mutations, but I think would be a hugely significant step forward in our efforts to develop novel ALS drugs. I just thought it would be useful, and I want to leave enough time for questions, to go through a little bit of the process, and Dr. Dave mentioned this in some of his schematics, but to give a specific example, our FUS ASO program began really in 2009 with the identification by, again, Dr. Brown and his colleagues and Chris Shaw, who found causal mutations in the FUS gene. And this subsequently led to the identification of dozens of mutations. This is a schematic just to show you that there are many, many mutations scattered across the FUS gene that are known to be responsible for the early development of ALS in patients with these mutations. And so in my lab, we began, based on this work, to study FUS ALS, as did many, many groups around the world. And we generated a number of mouse models to study this disease with the hope of assessing the consequences of these mutations on FUS activity and study their effects on motor neuron survival and function, but also to use them as models to test novel therapeutics like our FUS ASO. And this is just a picture from our mouse. This is a motor neuron expressing the mutant form of FUS, and you can see here that this graph just shows that our animals lose motor neurons over the course of their life in a way that qualitatively is very much like what we see in patients. So using this model, we were able to test J-diffusin, test our FUS ASO in a mouse model and demonstrate that it was able to lower FUS levels in the brain and spinal cord of these animals and have an impressive effect on the motor neuron phenotype that we saw that we believe was ALS-like. And based on this preclinical data, and there was others, safety data and other preclinical data that was provided by Ionis, we were able to move this therapeutic from the laboratory into the clinic first in a first-in-human expanded access program, and that experience and that demonstration of safety made, I think, it possible for Ionis to agree to move forward with this program and to sponsor Fusion, this phase 1/3 pivotal trial of J-diffusin, formerly ION 363, a global trial sponsored by Ionis Pharmaceuticals, for which we will be the lead site. And so I think I will maybe end there, but maybe just to say very briefly, and this is something that Dr. Dave touched on, there's a lot of exciting developments in the ALS field, too many really to touch on tonight, but you will hear tomorrow in the review of ongoing clinical trials in ALS, that there is an increasing focus on the role of neuroinflammation in ALS. And this is an idea that's been developing over time. This is an image from Dr. Appel's work almost 30 years ago, where he showed that there were lymphocytes, blood cells, that were infiltrating into the brain and spinal cord of ALS patients. And he subsequently showed in mutant SOD1 mice that these lymphocytes, these actually T cells, a specific kind of lymphocyte, directly kill neurons in a mouse model of disease. And he went on to show in a clinical trial that a type of regulatory T cell, Treg cell, that modifies the properties of these toxic, damaging T cells might be effective in ALS patients. And Dr. Appel now, in partnership with Koya Therapeutics, is moving this T cell work forward, and there are a number of other players in the field, including Rapid Therapeutics, which just initiated an open label study of their Treg cell, and work that we're doing with a company called Celenkos that has specialized in cord blood-derived regulatory T cells, is moving ahead in ALS. So there's a great deal of effort you'll see in active clinical trials to modify neuroinflammation, as well as to deal with the abnormalities that are happening within the motor neurons that are responsible for ALS. So two areas of great interest and promise in ALS, and I think there are many more of these kinds of studies to come. So I'll end there, and we have, I guess, 20 minutes or so left for questions, and I'd be happy to answer.
Thank you so much. Thank you, Dr. Shneider. And thank you, Dr. Dave, as well. I really appreciate these very informative presentations, and the fact that you have both used the word exciting numerous times throughout your presentations. I find to be wonderful, because as I probably mentioned to each of you, I do try to convey that message in my day-to-day work and my day-to-day conversations with the ALS community, because it really is an exciting time in ALS research. And Dr. Shneider, I want to congratulate you on the JC Fusion being accepted into phase 3. I think that's very exciting. Or fusion. I think I will always call it JC Fusion as it progresses through. But that's an amazing accomplishment. And I think it also, you had pointed out just a small sample of the sheer length of time that these things take to move through that extensive pipeline that Dr. Dave had referred to. I think that you showed a slide where something, it had been identified, or maybe it was the fuss that was talked about in 2009. And now it's coming into phase 3. So I think that it proves, that we need to keep moving this forward. I also want to thank Dr. Dave for showing that clinical trials, that very specific phase 1, 2, and 3 clinical trial slide. It is one of my favorites, and it does change that number of trials that are in that part of the pipeline. I think the first time I took some real notice of it, it was just not that long ago. And I think it was at 30. And here we are over 100, which is really just, again, incredibly exciting. And I want to thank you both for joining us today to give that update. Tiffany, I'm not sure if there are any questions in the chat that you want to start reading off.
Yes, thank you, Kristen. We have just a few questions here and have time for a few more if you'd like to go ahead and type those in. I'll repeat one that Dr. Dave actually answered online, but not everyone may be able to see it. And that is regarding the ALS perspective on the need for expanded access program for drugs in clinical development. To which he replied, every company needs to have an expanded access plan and should be clear about what that plan is. So Dr. Dave, did you want to say anything more about that?
No, I think that is our perspective. That and when we meet with companies, when they come to us or they inform us that they're going to start a trial, this is a question that we always put to them, whether they have thought about an expanded access plan, and they should make it clear to the general public what that plan is. To the ALS community what that plan is. And so, the example I like to give on that is when Amylyx reported positive data from their AMX35 phase 2 study, and we, along with IMLS, petitioned that Amylyx make AMX35 available through expanded access until it is brought to market.
Okay, great. Thank you. A couple of other questions here. I know that Kristen said we're all very excited to hear the word exciting. So you're not supposed to use the same word in a sentence twice. However, I'm going to. So what exactly do you think or what do you see as sort of the most exciting projects in development at this time? Do you have one or three favorites?
I guess I'll take that. You know, it's hard to point to a single favorite. I mean, it really depends. If you have a genetic mutation, the most exciting and promising, I think, programs are those which are specifically targeting your mutation, or the gene in which you have the mutation. So, but that's again a small minority of patients. So, you know, I think that the most exciting, I try to highlight the efforts in neuroinflammation. I think there's a lot of promise there. And I think that I didn't talk about it very much, but we're understanding more and more about the nature of the underlying pathology. That is, these proteins that I mentioned, these RNA binding proteins, there's a lot that's understood about how they behave in a cell, normally and in the context of the disease. And there are some very early events that I think lead to toxicity. And understanding those early events and developing molecules that might alter the behavior of these proteins in a way that might minimize their toxicity, or the formation of assemblies of proteins that are toxic. I think that's a very exciting approach, and there are a number of companies now that are focused on that effort. And so, I mean, in general, the therapeutics that interest me most are the ones that are based on a understanding of the disease mechanism, not evidence that I would say is less convincing, which is at some point all we had to go on. As we were learning more and more about the disease, we were able to make more rational decisions about therapeutic approaches. That's what's exciting at this moment, that the therapeutics that are coming out are based on an understanding of the disease, which we didn't have 20 years ago. And so I think that the chances of success are much higher, and that the history of ALS therapeutic research, which has admittedly not been impressive, that our future is much brighter, is I guess what I would say in the end, because we're taking a much more knowledge-based approach to therapeutic development.
Thank you. That actually leads into my next question here, which is a little bit multi-pronged. So maybe you could both take a stab at it. What does a cure for ALS look like? What does it look like? Is it living with ALS, or is it preventing it in the first place? Is it a combination of something like that? Where do you think we're headed in the next 10 years?
Neil, do you want me to take that? Maybe I can take a research stab at that, and then Dr. Shneider can provide maybe the clinical perspective. But it's tough to obviously. Maybe this is how I'll answer it. Obviously, we would like the cure to be something that stops the disease completely in its tracks. The issue right now we have is by the time somebody gets diagnosed, they've already had a loss of about 50% of roughly 50% of their motor neurons. And so how do we stop the disease right then and there? And that alone can be a big deal, because if we can stop patients from progressing, even if they're diagnosed with ALS, that's a big deal. And then the flip side, and I think inherent in that question was, can we prevent it? And that is really interesting, because if it's true that, and pathology is showing us that it seems to be the case that, by the time you're diagnosed, there's about 40, 50% loss of neurons. can we arrest that? Can we find biomarkers? Can we find clinical signs during the time when the pathology is already happening, but the symptoms haven't come on? And so, and this is the type of work that Dr. Michael Benatar is doing down in Florida, where he's looking at people that have the mutations, but the disease, they have not been diagnosed, what we call asymptomatic mutation carriers, and trying to understand clinically, genetically, biomarker-wise, what is different and what is the point when they do start to get symptoms, and can we detect that? And if we can, then maybe there is a way to arrest that prior to the disease setting in, similar to what we're doing with cholesterol right now, right? If you have an increase in cholesterol, your doctor puts you on cholesterol medications before you go on to get heart disease, or even if you do go on to get heart disease, having taken cholesterol reducing medications is going to help you with the disease. And so that's where we want to get to for ALS.
Yeah, I mean, I would agree. I mean, we too have a pre-symptomatic program. We call it the ALS Families Project, where we're following people with who are carriers of ALS mutations in the pre-symptomatic phase of the disease. And I think that there's increasingly good evidence from our model systems to suggest that getting in early could be preventative, meaning that is, if you could prevent the onset of the disease, that may be a cure, if you will, or at least equivalent to a cure. As Dr. Dave, pointed out, by the time a patient comes to us in the clinic, you are already in a difficult situation where you've lost probably majority of neurons, at least in the affected muscles. But ALS progresses regionally. It might begin in an arm and go to the leg, or go to the other arm, or it moves up and down the neural axis. And the hope is that you could intervene, potentially early enough to slow the spread and the progression of the disease. And many of my patients will say, Dr. Shneider, I can't use my right arm, but if you could just stop it there, I could live with this, right? I mean, and at each successive stage of the disease, they say, if you could just stop it here, I could live with this. So, I would say in answer to this that sometimes it feels too much at this moment to think about a cure. We're interested first in slowing disease progression and making a 3-year disease into a 10-year disease. We would ideally like to halt, ideally we'd like to halt progression of the disease and people know this term now, but bend the curve, and so that it is now, people don't progress further with a static deficit, and of course the real ideal is to actually have a recovery from functional loss, which is not inconceivable. The nervous system has some ability to heal and to compensate. We just need to, I think, put our patients in a position where that is possible. So, I think that we have a lot of interesting approaches now that are in, or soon will be in clinical trial, and I think that our expectations for these therapeutics are increasing, month by month, year by year.
Thank you. It looks like we have about just 5 minutes left. I do have 1 or 2 more questions, but if anyone else would like to go ahead and please ask a question, please go get it into the chat before we close out our session at 7:30. So we do have a question on veterans and, of course, understanding the connection between ALS and the military. There has been an increase in funding from the Department of Defense, who seems to be taking this very seriously. Can you speak on the connection, or what we understand at this time, and are we making any progress in this understanding this connection between military service and ALS?
Yeah, I can talk about this. This is a topic that is front and center for us. Remember, in my talk, I spoke about convening, and we convened a workshop in October of last year where we talked about its environmental and occupational causes of ALS. And we, in fact, had a military veteran who is now in ALS, who now has ALS, excuse me, and who also is on our board of trustees. He was at that meeting, and actually there were a couple of other ALS patients as well who also had military service, and you know, this is an important question because we need to understand why is it that they have an increased risk, a doubling, almost essentially a doubling risk of ALS. Is it their exposure to certain type of chemicals during their training or during their service? Is it, you know, toxins? Is it being around equipment? Is it being, you know, doing tours, military tours in other countries where they may be exposed to certain things? Is it PTSD? Is it stress? Is it chronic stress? Is it exertion, muscle exertion? I mean, there are just so many different things that could be driving that. And as Dr. Shneider said earlier, you know, we can't pinpoint one thing right now, but there are studies both that we're funding, that the CDC is funding, that the Department of Defense is funding, that is trying to correlate or link, you know, looking at military service, looking at exposures in the body, looking at, you know, trying to map out where that person may have served or in what capacity they may have served, and then linking it back to the diagnosis and when they were getting diagnosed or when they got diagnosed, excuse me. And so these studies are going to be very important to create that link. You know, Dr. Shneider went through this graph of linking genetic abnormalities. And in many ways, that has gotten easier as gene sequencing approaches have gotten easier. If we need to do a similar type of thing for environmental and occupational abnormalities, and how do they link up with the disease? So thank you for that question.
Okay, and that's perfect. That actually wraps up our time this evening. I want to thank Kristen Kokeman, Dr. Shneider, and Dr. Dave for answering our questions and being available. We tried to answer everyone's questions, and if we didn't get to your question this evening, we will follow up with you. Please attend the wrap-up session with our chapter staff on Friday at 3:45, and we'll answer any of your outstanding questions. I hope you all have a wonderful evening, and our session starts tomorrow in the afternoon. Check out the agenda for our full scope of sessions. Thank you.